Hey guys, we're going to pick up where we left off on our notes. And so I'm not sure, I kind of put a message in there. If you guys didn't finish your big model yesterday, you can work on that and then we can move on to these notes. But let's go ahead and finish up our packet, kind of see how far we get today. So I think we left off with white matter and gray matter. And so we were talking about functional versus structural neurons. And so functional or sensory or motor interneurons, and structural would be like bipolar, unipolar, and multipolar. Okay. And so functional is telling you what they do, and structural is telling you what they look like. And so let's take a look at what they can look like. Bipolar is this top one. This is kind of what we've seen a little bit. You have an axon or dendrite, you have the cell soma, okay, the body, and then the axon, and then the axon terminals. A unipolar neuron, you can notice that the actual cell soma is a little different. It's kind of extended above with kind of axons coming out. And then here we have like our multipolar with our dendrites coming into our cell with our collateral branches. Some fun facts about neurons. They can live and function your whole lifetime. That's why when they talk about doing things that kill brain cells, it's dangerous because nerve cells can live your entire life if they're allowed to. They don't divide. Fetal neurons lose their ability to undergo mitosis. The only exception there are stem cells. So there are neural stem cells that'll keep dividing. So you can make new ones, but not that many. And that's why nerve damage is so dangerous and why it's so serious, because unlike all our other cells, neurons really don't go through mitosis. They have a very high metabolic rate, so they need a lot of oxygen, doing a whole lot of cellular respiration. And so neurons are going to need a lot of oxygen, a lot of glucose to keep them fed. And that's why malnutrition can affect your brain and your nerve function. The nerve fibers of babies are unmyelinated, and that makes their responses really coarse. And that's why, like, they have jump reflexes much more than older babies will, and just kind of react different. That's part of what they test for, in fact. As the baby develops, that kind of changes as their sheaths become myelinated. Now let's talk a little bit about cell membrane potential, okay? So in this, basically a nerve impulse is a weak electrical current. It's like a wave running down the length of the nerve. And so you have the resting potential, the threshold potential, and the action potential. And this is just kind of a little image showing how things move across the cell membrane in and out of the cell, causing this impulse to move down like a wave. The neuron membrane maintains the resting potential, okay? And you can see here that we have these voltage-gated sodium channels, and they're closed at the resting potential. Channel's closed. Then the threshold stimulus is received, and the sodium channel opens, okay? And so basically the nerve impulse makes this gate open, and sodium comes across the cell membrane. Sodium ions diffuse inward, depolarizing the membrane. The potassium channels open, and potassium moves out. This is the sodium-potassium pump, it's called. And so this change of sodium-potassium is what changes this charge along that cell. The resulting action potential causes a local bioelectric current, so just a little, really, electrical current. And that wave is an action potential, travels the length of the axon, and that's the nerve impulse moving down the nerve. Okay, so there it goes. Sodium, potassium moving, causing the wave of action, and that's carrying the message down to the next nerve cell. There it goes again. The resting, and here's just kind of another image of it. This is in your notes too. The resting potential, right? Sodium, potassium. Stimulus initiates the local depolarization and generation of that current. And you can see that sodium moving in. okay? And that action potential moving. Ions in the cell and outside the cell create a positive and negative side, which produces an electrical current, just like a battery, okay? So here's a simple battery circuit with a positive and negative side. And so very much the same idea, okay? So the current you see in a battery, the current you see in a nerve cell. Depolarization is the loss of the difference in charge between the inside and the outside of the membrane of a muscle or nerve cell due to a change in permeability and migration of sodium ions to the interior. And this is basically, you've got the depolarization, repolarization, back to the threshold, right? So resting, action, resting, okay? And here's just another image of that and how quickly that can happen. The speed of a nerve impulse is proportional to the size of the axon. So the bigger the axon, the faster the impulse. Myelinated axons conduct impulses faster than unmyelinated ones. And that's why babies' responses are different and kind of completely different from ours. So you can see that axon passing that signal down to that neuromuscular junction. The synapse, okay? There was a little bit about this on your assignment from Monday, and let's kind of go over it. The synapse is the junction between two neurons. Neurons don't touch. There's a gap between them, and that gap is the synapse. The nerve pathways, nerve impulse travels from neuron to neuron, okay? So what that means is we've got to go dendrite, cell body, along the axon, synapse, dendrite, okay? So synapse, dendrite, cell body, along the axon, synapse, dendrite, okay? And that's how the nerve impulse passes from nerve cell to nerve cell because they're not touching. That's different from other cells in our body. To complete the signal, a neurotransmitter is released into the gap to signal the next neuron. Receptors on the dendrite receive that chemical message. And so these little guys right here, there's the synapse, the gap between the two. Here's a little synaptic vesicle, which is holding the actual neurotransmitters. And these little black dots are the neurotransmitters going across that gap to the receptor of the next nerve cell. Notice there's some mitochondria in here. Okay, it takes a lot of energy for this to happen. So here's really the anatomy, okay? So A is a neuron, the axon end. B is a neuron, the dendrite end. We have mitochondria. We have these little vesicles, which are just little pockets of neurotransmitters. We have little receptors on both ends, okay? The synapse, the gap. And here's the receptors on the receiving end. Here we have a calcium channel because calcium is going to play into how this all works. 7 is showing the release of the neurotransmitters, and 8 is showing the reuptake, okay? And so this is all going to play into how one message jumps the synapse. There are 2 types of neurotransmitters. They're excitatory and inhibitory. Excitatory increase the membrane permeability, increase the chance for the threshold to be achieved. Inhibitory decrease the membrane permeability and decrease the chance for the threshold to be achieved. So you can actually have synapses that cause the message to keep going and synapses that cause it to actually slow. Examples of neurotransmitters that you have probably heard about before, okay? Some of these. Acetylcholine is the big one, and that's the muscle contraction neurotransmitter. And we already talked about this one when we talked about muscles. So acetylcholine, that's our big muscle contraction neurotransmitter. Dopamine, this is a happiness neurotransmitter. You hear about dopamine being good for your mood, which is true. Okay, so it's the neurotransmitter that kind of sends good vibes. Serotonin, this is the nice sleepy one. Okay, you might take some kind of serotonin to help you sleep at night. It also helps with mood. And so serotonin is actually a neurotransmitter. Endorphins, we've definitely heard of these. Endorphins are pain reduction and help our mood. And so all of these things, endorphins, serotonin, dopamine, when you hear about them, what they really are in the chemical world of your body are neurotransmitters that are going between these nerve cells. Let's talk a little bit about agonist versus antagonist. Agonist molecules are that they have the same effect on the postsynaptic neuron as the neurotransmitter itself. And so an agonist is a drug usually that can replicate the neurotransmitter. Say you're not producing enough. Antagonist is a molecule that blocks the effective neurotransmitter. And so maybe you're producing too much of a neurotransmitter and you need to block some of it. So there are actually drugs that occupy that receptor, and you can see it here, and they can either partially occupy it or completely occupy it and keep those messages from being passed. Both of these can be effective treatments for different things. And this is a video about the science of depression. And so I'm going to go ahead, I'm going to actually link this video so you can watch it separately from this presentation. And I think depression is something important. A lot of people deal with it. And a lot of people maybe even in this class may have dealt with it or have family members who deal with it. And so let's talk just a little bit about antidepressants. Zoloft is one example. It's a selective serotonin reuptake inhibitor, okay? And it inhibits the uptake of serotonin back into the cell, keeping your mood elevated for a longer time. And so basically, normally when you produce serotonin, your body retakes it up after just a little bit of time. Zoloft blocks the reuptake, and so you end up with more serotonin that you naturally made staying and going to the next cell, okay? And so you actually have that elevated mood longer than you normally would. There are other drugs that can affect nerves. Curare is a poison made from frog skin, and it causes paralysis by blocking ACK receptors at the neuromuscular junction. And so poisons that cause like paralysis usually are blocking a neuromuscular junction, which is the case of like the poison dart frog and how it works. Strychnine, another common poison, it's rat poison. It can be fatal to other animals too. What it does, it prevents the proper operation of the chemical that controls nerve signals to the muscles. The chemical controlling the nerve signals works like the body's off switch for muscles, and when this off switch does not work correctly, they have like spasms, and so that's what strychnine does, and it affects that neurotransmitter complex as well. We're going to get a little bit into like illegal drugs, and there are some others we're going to look at. I have a really cool kind of activity we'll do when I get back, where we look at how these drugs affect people. Cocaine, for example, it's dopamine binds to receptors. Normally, dopamine binds to receptors and is eventually recycled back into the neuron, okay? And you can see that here where the dopamine binds to receptors and then it's recycled back into the neuron. If cocaine is present, it attaches to the dopamine transporter and blocks the normal recycling process, resulting in a huge buildup of dopamine in the synapse, which is where like the pleasurable effect of cocaine comes from. And along with that, obviously, the lack of cognition to make good decisions, because these stay open and it changes your interpretation of the world around you. And so cocaine actually attaches to those neurotransmitters and blocks it from coming back up the way it should. And here's just a picture of your neurotransmitters on cocaine. Here they come. And that would be normally what would happen. But the cocaine blocks the reuptake. And so you end up with a much, much higher dose of dopamine in the synapse than you should. And so it keeps sending that same signal when that signal should begin to lessen. Another drug, ecstasy, okay? The drug ecstasy deals with serotonin. And serotonin does a lot of things in our bodies as far as maintaining homeostasis. It makes us feel good, okay, but it also regulates our body temperature. And our brain cells recycle serotonin back into the cells and out of the synapse using serotonin reuptake transporters. Ecstasy essentially takes those upkeep transporters and reverses them and causes a massive flood of serotonin from the brain into the synapse. Well, because serotonin is in charge of regulating your body temperature, people who die from ecstasy die of overheating because your body it won't maintain your body temperature anymore because there's way too much serotonin being pushed into your nerve endings, causing your body temperature to rise. And hyperthermia, which is death from overheating, is what is a main killer from ecstasy. Heroin. Basically, heroin activates opiate receptors and blocks the release of GABA, which is what would kind of control the dopamine production. Okay. And here's kind of a picture of that. This would be normal, and this would be with heroin. And so what it does, it basically triggers that cell to release a whole lot more than it should, which in all of these cases, the issue becomes your body is out of any kind of homeostasis, right? And we know that we just can't survive indefinitely in a state of non-homeostasis. So you can see the heroin-induced versus the normal. Amphetamines. Basically, amphetamines themselves mimic dopamine. So it's not so much that they are doing, like actually changing the way your nerves act. They're like little fake dopamines. And so what ultimately happens is dopamine doesn't come back in because it's got this amphetamine mimicking it. And over time, you don't actually have as much dopamine as you normally would. And that's why coming off of amphetamines are very, very hard. Because your body's chemistry actually changes. So mouse party is what we're going to do when I get back. I think you guys will really enjoy it. Let's talk a little bit about impulse processing. You have these things called neuronal pools, which are groups of neurons that make hundreds of synaptic connections and work together to perform a common function. And really, pools are kind of what help us remember sequential tasks, things that we don't forget how to do. We don't have to reteach ourselves to do. So neuronal pools are things that help you remember how to tie your shoe once you know it, how to ride a bike once you know it, how to hold a pencil, how to write, even writing your alphabet and things. You have this neuronal pool that's actually kind of, it's not just I think we hear muscle memory, but it's really nerve memory here. That these pools help us kind of carry out tasks that we carry out without even having to think usually about it. It just kind of happens naturally because of these neuronal pools. Now, an actual nerve pathway, and we'll do some labs with this when I get back to a true reflex arc. If you guys have ever gone to the doctor and they've hit your knee with the little paddle for a knee-jerk reflex, this is a simple reflex arc, okay? It only involves a few neurons, really. It's involuntary, it's instant. And basically then, like, for example, the nerve jerk reflex, it maintains your uprightness. It's what keeps you standing up. The reason we have it is because that's kind of what helps us walk. Withdrawal reflex avoids the pain of the stimulus. And so it's there to keep that foot kicked out and keep you standing. But when you actually test it, you'll have that removal because of that painful stimulus. Even though it doesn't really get all the way to your brain understanding it, it's more of a spinal cord type of reflex. That's why it's so fast. A few disorders, and you guys looked up a few of these, and I'll link these videos as well. Lou Gehrig's disease amyotrophic lateral sclerosis, or ALS. It's a progressive degeneration of nerve cells in the spinal cord and brain. That means the symptoms only get worse. Patients die usually within 5 years of diagnosis of ALS. You may know someone who had ALS. Became very famous when a baseball player named Lou Gehrig died of it. And that's where the second name for it comes from. And I'll post a video about that as well. Epilepsy. Epileptic seizures are caused by excessive electrical activity within the brain. A fine balance between excitation and inhibition must be maintained for the brain to function normally. If there's too much glutamate, neurons can become hyper-excitable, and a seizure may result. And sometimes too little can result in a seizure too, and so it's a balance. And people with epilepsy have to be very careful about outside stimulus because they're constantly kind of trying to maintain their internal body stimulus, but sometimes outside stress or pain or even like lights and different things can trigger seizures for people with epilepsy. Myasthenia gravis is an autoimmune disorder where your actual antibodies, like your immune system, destroy your neuromuscular connection. Okay. And we've kind of talked about this one before, but it'll actually cause the destruction of this connection between the muscle and the nerve. And then MS, multiple sclerosis. Basically with MS, this is autoimmune as well, where your immune system attacks itself. Myelin around the nerve cells lost and forms scar tissue called sclerosis. And because there's scar tissue on those nerve cells, they can't conduct the impulse to and from the brain. Ultimately, it can affect your ability to walk, talk, swallow, and affect vision. But it's really the damage to that myelin sheath in those cases. And you can kind of see that in this image. And guys, I think that kind of gets us to the end of the actual notes. It'll probably be that for today, and tomorrow I'll post the videos that go along with it. And I'll probably post some like ed puzzles to get you some points on those. Once you have all your muscle or all your nerve notes filled out, make sure a lot of you uploaded the first half, but go ahead and upload the second half as well. And then we'll have the videos to go along with it. So hope you guys are having a good day and hope to see you soon.